Hi, I'm Arula Ratnakar, and this is my presentation, Architecture, Neuroscience, and Speculative Fiction, Relationships and Intersections. Hello? Okay, I heard a noise. And yeah, I have background in architecture, neuroscience, and speculative fiction, so I'm going to, I guess, just like get right into it because I have a lot to present, and I'll try to get through it all and finish in time. Oh, and I'm in Superton A for the Discord. I will be after I finish this because I can't do the Discord while I'm presenting, obviously. Okay, so I'll start now. Okay, so architecture, neuroscience, and speculative fiction. So for as long as we've been recognizably human, we've been interested in exploring the limits of our imagination, whether that's through creating awesome spaces in reality, imagining what could be possible if some aspect of our reality was changed or added, or dissecting and theorizing about the biological substrate of imagination itself within our nervous system. So all three areas of study are interested in how we perceive reality and process sensory information, as well as what controls our emotions and how our emotions can change, as well as what sticks in our memory and how memories form and how we move through spaces and select actions at any given point. So in this presentation, I'm going to walk through how each field, architecture, neuroscience, and speculative fiction, study these aspects of human experience through examples and through a lot of science communication. So I'm going to start with neuroscience and how we biologically process the reality around us through sensory information as well as how our memory works. I think that's kind of like the bare bones of what constructs our reality is like sensory stuff and memory. And then I'll walk us through architecture and how architectural design is kind of studying the same thing but from a different angle. And then after that, I'll go into speculative fiction and give examples of how architecture and neuroscience are kind of like combined through speculative fiction and how it in itself is manipulating our perception of reality. So yeah, my slides are going to be kind of a hodgepodge of like architectural pictures I took, and at the beginning for the neuroscience part, I'm going to use slides from the class I teach at Carnegie Mellon University, neuroscience for non-majors. So yeah, I'll start with our senses and how they work, beginning with vision. So the world as we see it is a result of cells in our eyes processing photons. And so this is a human eye. And when light hits our cells in our retinas, they're absorbed by photoreceptor cells called rod cells and cone cells. And rod cells contain a protein called rhodopsin, while cone cells contain a protein called photopsin. And these proteins are what process the photons arriving at the retinal cells and cause the retinal cells to fire an action potential, basically causing them to generate an electrical signal and cause a release of chemicals called neurotransmitter that can generate more electrical signals than other neurons in the presence of light. While rods are 1,000 times more light sensitive than cones, rods do not produce color vision and are primarily responsible for identifying presence and absence of light, black and white. Cones, on the other hand, do produce color vision, and there are 3 types of cone cells, either able to process red, green, or blue wavelengths of light. The combination of activations of these cones allows us to see the variety of colors that we do. And so these retinal cells activate cells in the optic nerve, which takes this visual information into the brain. Also, I'll take all questions at the end after I finish the presentation, but yeah, I'm gonna go through the presentation first. So when eyes take in information from their surroundings, the right eye's representation of the left half of the world, along with the left eye's representation of the left half of the world, are sent to the right hemisphere of the brain to be processed, while the representations of the right side of the world in either eye go to the left hemisphere of the brain to be processed. This information goes into a part of the brain called the thalamus, into an area called the lateral geniculate nucleus, or LGN. Each cell in the LGN is monocular, only processing information about the world that it receives from one eye or another, but not both eyes at once. The thalamus itself is a gate for sensory information and processes the information before sending it out again into appropriate areas of the cortex being the outermost layers of the brain. After making it through the LGN, visual sensory information is sent to the fourth layer of primary visual cortex, or V1, which is located in the occipital lobe. The entire occipital lobe, parts of the temporal lobe, and parts of the parietal lobe are all dedicated to vision, combining vision with audition or combining vision with somatosensation. And layer 4 in primary visual cortex, or V1, has monocular cells. From layer 4, it goes up to layers 2 and 3, where the interconnectivity allows for binocular cells that can compare information from both eyes within an individual cell. Binocular cells are very important for judging locations and distances of objects, as they compare positions of objects in one eye to position of those objects in the other eye. Layers 2 and 3 project down into binocular cells in layers 5 and 6, and the projections from layers 5 and 6 lead out of the primary visual cortex through two important streams of information, called the dorsal stream and the ventral stream. So these are the lobes of the brain, and the ventral stream projects into the temporal lobe and is used to identify objects, animals, and people by combining information about shape, color, and sound. However, faces have a separate area called the fusiform face area on the bottom of the temporal lobe, where they're represented and recognized. The dorsal stream flows into the parietal and then to the frontal lobes, and it carries visual information about the location and motion of objects, putting together how an object can be interacted with, as well as where it is and how it is moving. This is why it goes to the parietal and frontal lobes, because it needs to combine visual information with somatosensory and motor plans, and that's where somatosensory and motor cortices are. Okay, on to hearing. What we hear in the world is a result of how we process sound waves around us. First, sound energy is gathered by our external ear into the tympanic membrane, our eardrum, especially amplifying sound frequencies that include our most important speech sounds. Then the pressure on the tympanic membrane is focused onto a place called the oval window, the entry point into the inner ear from the external ear. The cochlea of the inner ear is responsible for converting the sound energy into neural impulses. It's a coiled up structure that has three fluid-filled chambers called the scala vestibuli and the scala tympani, which contain a fluid called perilymph, as well as the scala media chamber, which contains the fluid called endolymph. When the oval window is hit by a sound wave, these fluids are disrupted, and the fluid movement causes a membrane called the basilar membrane to move in a wave-like manner as well. Different parts of the basilar membrane respond differently to particular sound frequencies. When this wave movement reaches its optimal location in the basilar membrane, it disrupts these things called hair cells in that area that sit just above the basilar membrane. On these hair cells, microscopic hairs called stereocilia are disrupted, and this disruption opens ion channels in the hair cells that allow the cells to generate an electrical signal and release neurotransmitters, which then takes the auditory information into the brain to be processed. And over here is the basilar membrane, and even though it's coiled up like this, it's just easier to visualize like this. In a part of the brain called the medial superior Oliver MSO, interaural timing difference is computed by having cells that receive inputs from both the left ear and the right ear arranged so that longer axons, where axons are projections of neurons that propagate an electrical signal, are coming from the left ear's cochlea, coming from the right ear's cochlea are paired with shorter axons from the left ear's cochlea and vice versa. These MSO cells only activate when they receive inputs from both ears' axons at the same time. And that's how you know where a sound is coming from. For example, in this diagram, when a sound arrives at the left ear first and the right ear second, the longer left axon will be activated and the shorter right axon will be activated so that the simultaneous inputs will arrive at neuron E, which represents a left ear leading sound. If the simultaneous inputs arrived at neuron C, which has equal length axons from left and right ears, it would mean that the sound comes from directly in front of the person. If the simultaneous inputs arrive at neuron A, it would mean the sound is coming from the right. The lateral superior olive computes interaural intensity difference, and it's less complicated. Basically, the side of the LSO that's the same side of the head that the sound sources is on will be more excited, and the opposite side will be more inhibited. The information from the MSO and the LSO goes to the inferior colliculus in the midbrain, and then goes to the medial geniculate complex in the thalamus. From the thalamus, it goes to the auditory cortex, which contains a topographical map of the parts of the cochlea, and then it can be integrated with other information from the brain. Okay, I'll go over olfaction and somatosensation, but I'm going to skip over taste and pain and the neuromuscular junction for the sake of time, because I want to make sure I get through everything. So we smell because of particular chemical combinations that activate particular receptor combinations in our noses. And these chemicals are a wide range of airborne stimuli called odorants, and they interact with receptor neurons on the olfactory epithelium, which is a lining on the interior of the nose. The axons from these receptors project across a region of the skull called the cribriform plate into a part of the brain called the olfactory bulb. Neurons in the olfactory bulb project to a cortical region called the piriform cortex in the temporal lobe, and these projections, called the olfactory tract, are the reason why smell and episodic memory are highly linked. The temporal lobe contains the hippocampal formation, which we'll learn about in a moment, which is crucial for memory. The olfactory information goes through the piriform cortex to the thalamus, and then it goes to targets like the hypothalamus, which controls hormone secretion, and the amygdala, which controls fear response, as well as other areas of cortex. Okay, now the somatosensory system is responsible for the sense of touch and everything associated with it: pressure, temperature, pain, itchiness, etc. It has three main components: one that transmits information about touch, vibrations, and pressure, a second subsystem responsible for proprioception, or our ability to sense muscle, tendon, and joint position in space, and a third subsystem conveying information about pain and temperature. The skin has different amounts of sensitivity depending on its location. For example, on your fingertips, you can identify 2 points of contact if they're 2 millimeters apart, but on the upper arm, you can't perceive two different points of contact as being distinct until they're at least 40 millimeters apart. Neurons that receive information from the skin and other organs are considered afferent neurons, while neurons that send information out to the organs are called efferent neurons. So rapidly adapting afferent neurons, called Meissner afferents, are all over the hand, and they fire rapidly when a stimulus is first pressed against the skin, but then fall silent if the stimulus stays there. Slow adapting afferents, called Merkel cells, which are in the fingertips, generate a sustained response with a continuous stimulation. These skin sensing, or cutaneous, nerve cells are called mechanoceptors and open ion channels in the presence of pressure or distortion of the skin. Meissner corpuscles are really good at conveying information about textured objects that move across the skin, while Merkel cells are able to identify distinct stimuli even if they're very close together. Pacinian afferents are also there deeper in the skin, and they are rapidly adapting and are good at sensing high-frequency vibrations. And Ruffini afferents, on the other hand, are slowly adapting and respond well to stretching of the skin. So all of these have cutaneous receptors and provide information about external stimuli. Proprioceptors are a different class of mechanosensory neurons that are responsible for giving detailed and continuous information about position of limbs and other body parts in space. For example, joint receptors in and around finger joints activate when a finger joint reaches its limits of motion range. Sensory afferents are also wrapped around central fibers of skeletal muscle spindles and open ion channels when the muscle is stretched. The muscle spindle is innervated with two kinds of nerves, primary and secondary. Primary nerve endings transmit information about velocity and movement direction, while secondary endings provide information about static limb position. Cutaneous and proprioceptive information travels through the dorsal horn of the spinal cord and up the ascending spinal cord pathways to the brain. In the brain, the information travels through the pons in the brainstem and midbrain and ventral posterior thalamus to the primary somatosensory cortex, which contains a topographic map of different parts of the body. Your motor cortex, which is right next to primary somatosensory cortex, also has a topographic map of different parts of your body. From the primary somatosensory cortex, information goes to the motor cortex or to the secondary somatosensory cortex. And from the secondary somatosensory cortex, it would go to areas like the amygdala and hippocampus, while from motor cortex, it would go to like your brainstem and into motor neurons, and motor neurons directly synapse onto muscle cells, causing contraction in the muscle cells through an electrical signal. Okay, I'm going to briefly go over how memory works, and then I'll move on to architecture. The hippocampus is a structure that, and this is just, I'm going to go over short-term memory, and it's like episodic and contextual memory, which is like more limited than I'd like to talk about, but I think it's the most important structure for memory. Episodic memory is the hippocampus, but just know that during sleep, short-term memory is actually transferred out of the hippocampus and into cortical storage sites again. And that's the process of turning short-term memories into long-term ones. So yeah, the hippocampus is a structure that is embedded in the temporal lobe, and we have two, one on either hemisphere. It's divided into different parts and connected to a structure called the subiculum, which is connected to a part of the cortex called the entorhinal cortex. The parts of the hippocampus are the dentate gyrus, which contains a lot of neurons, and the cornu ammonis, or CA. The dentate gyrus is thought to be one of the only places where new neurons are actually born in adulthood, neurogenesis. Other than the dentate gyrus and I think another part of the brain called the striatum, the neurons are baby-like neurons aren't being born throughout your brain, other than those two areas. So cornu ammonis, meanwhile, is divided into 4 subfields as shown. And these structures are all crucial for both memory and navigation. So there are specific names for the different axon projections between various parts of the hippocampus and entorhinal cortex. The projections from entorhinal cortex to dentate gyrus are called the perforant path. The projections from dentate gyrus to cornu ammonis subfield 3, or CA3, are called mossy fibers. And the projections from CA3 to CA1 are called Schaffer collaterals. In terms of navigation, the entorhinal cortex contains grid cells, which fire at regular intervals throughout the process of spatial navigation and create a sort of grid map of the space you're navigating. This is occurring alongside a process in the cornu ammonis, which allows you to pinpoint your location within that grid map. Cornu ammonis subfields 3 and 1, CA3 and CA1, contain pyramidal place cells. These are spatially localized cells, meaning a portion of them will drastically increase their firing rate if you occupy a specific location, and a different portion of them will drastically increase their firing rate if you occupy a different location. So this specific location that triggers high frequency firing in a portion of the CA1 and CA3 cells is called the cell's place field. So memory can be divided into 2 main categories of declarative and procedural. And declarative memories, which we'll focus on for today, is memory that deals with conscious recall of facts and past experiences. It's the explicit memory. Procedural memory, on the other hand, is implicit memory and does not require conscious recall. So the mechanism by which short-term memories form is called hippocampal indexing. So during an experience, all sorts of areas all around the brain, like the ones we talked about before we began this discussion on memory, are active, right? So these areas are all sending inputs to the entorhinal cortex during an experience. All of that like sensory information that we're talking about, all of this stuff that makes up the snapshot of a moment in time that you're experiencing is sent to entorhinal cortex and during an experience. And if the experience is going to be stored as a memory, then the entorhinal cortex will activate a unique group of neurons in the dentate gyrus through the perforant path, as the entorhinal cortex is getting all of those inputs from all the areas of the brain that are currently active. And the dentate gyrus has enough cells that it'll be able to activate a unique combination of dentate gyrus cells for every experience. This unique group of cells corresponding to the experience is called an engram. And the dentate gyrus engram activates another engram in cornu ammonis subfield 3 that corresponds to it through the mossy fibers. CA3 is special because it has a lot of reciprocal projections within itself. So all of the neurons in CA3 are connected to each other, both as presynaptic, like so the ones sending neurotransmitter, and postsynaptic, so the ones receiving neurotransmitter neurons. Okay, so now we have an engram in CA3 from an engram in dentate gyrus from activation in the entorhinal cortex. Now, if a portion of the original experience is present sometime later, which is what a reminder is, then that portion of the brain will reactivate a tiny portion of the entorhinal cortex cells that were active during that experience, which will activate a tiny portion of the dentate gyrus engram, which will consequently activate a tiny portion of the corneomonas subfield 3 engram, because of all of those reciprocal interconnections, which will consequently reactivate the entire engram in CA3. This full engram now activates corresponding cells in CA1, which activates corresponding cells in the subiculum, which connects back to entorhinal cortex and reactivates all of the entorhinal cortex cells that were active during the original experience. Now, because of reciprocal projections from entorhinal cortex back to all of those various areas in the brain, this activation at entorhinal cortex, triggered from projections from the subiculum, will now go and reactivate all of those same areas of the brain that were active during the original experience. And this is a memory. It's super cool. And yeah, like, one thing that I love about the hippocampal indexing circuit is that it kind of reminds me of almost like an encryption algorithm, and like how you need this sort of unique key, this like unique set of cells in dentate gyrus to basically reactivate this sort of like or to like I don't know, to reactivate this message or something. I don't know. I feel like there are parallels there. There is so much more that goes into how our brain constructs the world around us, but I hope this gives you all a general idea. And these days, neuroscientists have a great deal of tools at our disposal. Neurophotonics, in particular, is something I'm interested in. Neurophotonics is the use of optical technologies to study the brain, and it's a super exciting field. We can actually genetically modify neurons to contain light-sensitive proteins and use this to either, visualize neural firing through this process called like calcium imaging. Of course, this is in rodents. Or if the light sensitive proteins are ion channels, we can actually direct light onto cells to actually trigger neural firing or inhibition in a process called optogenetics. So I'm actually going to go into neurophotonics after I graduate in May, which I'm really excited for. I personally believe in just inventing whatever tools we need to study the questions we want to study if those tools don't exist yet. So optogenetics also actually features heavily in my latest hard science fiction novella, Submergence, which you can read in the March 2021 issue of Clark's World. But anyway, neurophotonics is one exciting field that manipulates these circuits to interrogate and study them so that we know what goes into constructing human experience. Neuroscience, at the end of the day, is what studies and deciphers the foundation of human experience, which is why I started with it. And it's the most in-depth part of my presentation. Architecture and speculative fiction build onto this foundation and manipulate these circuits and processes and play with them and turn them into art. So navigation, smell, sight, sound, and textures of the world around us are what creates snapshots of our experiences that turn into memories. And our strings of unique episodic experiences and memories are what change our neural circuitries on an individual level and make us unique. So when we create a space through architecture, utilizing those sensory stimuli, we are contributing to this individual synaptic plasticity and shaping of personality and identity for every single person that walks through a space. The most salient episodic memories are formed from the richest sensory environments. So it is so important to think about the way like light will fall onto the occupants of a space or onto the textures of the walls and floor. Textures that people will like lean against or sit against or brush their fingers against. And it's important to think about the sense you will experience as you walk through a space, like cedar wood or grass or rainwater. And it's important also to think about like echoes in an auditory environment around you in architecture, how sound carries from one end to another of a room. And in architecture, you can also play with your neural circuitry. You can actually, you can use physics to your advantage in architecture and design ways that like sound will carry through a space, for example, or like design and like trick your visual system into thinking there are like infinite of something when there's only one, using mirrors, for example. You can really like play with what you see, and you can really like trick, ultimately like your experiences are just how your brain is processing different sensory information and integrating it. So you can really like use that to your advantage and kind of like trick your brain's processes into creating like the like most vivid and amazing sensory experiences out of like the bare minimum materials and resources. So you can, curate that place cell activation sequence in your hippocampus through how you choose to navigate a space. The best architecture in the world will make you feel as though you're choosing how to circulate through a city or like a building yourself, when in reality, every single step of your path has been carefully crafted and calculated by the designer. Often, when I used to be an architecture student, and often in architecture school, we were asked to start with an interesting path, just a path, and then construct the space around that path in a process called circulation diagramming. Actually, I recently got another novella accepted by Clarkesworld. Just last week, I got an acceptance, and it's called Babirusa. And there's imaginary architecture in it, and I used this very process of starting with an interesting path and designing a space and a world around it to come up with some pretty cool imaginary architecture. And so, yeah, navigation and circulation through a space is super important for both memory curation, because again, remember, navigation is the same system that encodes your episodic memories, yeah, as well as speculative fiction and things like that. So architecture is the study of contexts and spaces, and whether man-made or natural, it's kind of the study of your environment. And the reason your brain can make up a reality for you, the source of all your experiences, the main driver of your very identity, the catalyst for your every relationship and interaction. I mean, like, how could you have an interaction without a space to hold it in the first place? Like, even now as we speak, your memory of this moment will be shaped by your current unique surroundings and the objects within it, providing sensory signals for your brains to process. But architecture does have its limitations like physics, like building code, like budget. Speculative fiction does not have these limitations. It can toy with your mind in a way that real world architecture is not able to. From the famous labyrinth in the myth of the Minotaur to the infinite library of Babel in Jorge Luis Borges' story, to the endless, repetitive, hypnotic architecture of Piranesi, we have used speculative fiction throughout history to break the laws of physics and design and construct environments that would not be able to exist in reality. The retrosplenial cortex in the temporal lobe, right by the hippocampus, is what helps drive imagination of fictitious events. But the hippocampus and all the neurocortical storage sites that memories go to when they become remote memories, a process called consolidation that happens during sleep, are important as you read a story. You're consciously activating and reactivating sensory processing areas and memory areas of your brain as you imagine and visualize a story that you read. As writers, you are also architects of the imaginary realm, kind of inventing sensory environments for your readers. When you describe meals and settings and sensations and sounds, you're also actually shaping the identities and personalities of your readers, adding to these snapshots of experience being processed through their hippocampus and cortical areas, driving synaptic plasticity to implant memories and experiences into their skulls. All art can do this, but fiction can actually do this with an exactitude and a level of resolution and detail that is unparalleled. You do not need to decouple reality from the senses, like with real architecture, to toy with readers' brains in speculative fiction. You can instead invent whatever rules and whatever reality you want, and actually write the sensory signals to match. Yeah, and that's it. Thank you. Okay, that actually went way faster than I thought it would. When I practiced, it was longer. So I guess I have time for questions, I guess. I hope that went okay. Or am I supposed to go to the Discord, or? I guess there aren't questions. There's like a chat here. But yeah, that was it. And yeah, I guess maybe I should say that I started out in architecture, and then I went into from architecture, I got into like set design, and I wanted to be a set designer for film. And then I went into science fiction writing and screenwriting because of things I liked in learning like set design and film. And that led me to like actual science. So that was kind of my journey through undergrad. And I'm going to graduate this May. And I guess you can find my published science fiction stories. I have 3 of them published and 1 accepted, all in Clarkesworld magazine. And yeah, I definitely talk about like these kind of themes and things like that in my stories. Yeah, and then I'm going to go into neurophotonics. There's a chat here, but I'm not getting messages in this chat. So I guess I'll leave and go to the Discord instead.